Okay, so we're going to talk a little bit about the lytic and lysogenic cycles, specifically for bacteria cells. And then in a couple of minutes, we're going to talk about a third cycle that we sort of just touched upon, but we'll go over it a little bit more formally, that we find in animal cells. So over here, you can sort of get a little preview about how an infection could lead to the lytic cycle or the lysogenic cycle, also known as lysis or lysogeny. So we'll start with the lytic cycle, which is probably the most well-known in mainstream in pop culture, especially with the rise of the COVID-19 pandemic. You start to see a lot of information about viral transmission, viral life cycle. And in terms of COVID-19, we're usually talking about the lytic cycle. So after the virus attaches to a cell and inserts its genome, it can enter one of two cycles. The first of which that we're going to discuss is known as the lytic cycle. So when the virus enters a cell, it will usually start expressing genes that encode for an enzyme known as hydrolase. So this is basically the first thing that the virus does when it enters the cell. So because this gene is expressed as soon as the virus enters its host, it is known as an early gene. So what hydrolase does is it breaks down the cell's existing genome so the virus has access to building blocks to replicate its own genome. So a bacteriophage, for example, is a DNA virus. If it needs to replicate its genome, what better way to do it than with the building blocks of the own cell, of the host cell? Because in the lytic cycle, it's sort of like scorched earth. We're going to take this cell, we're going to take everything that we can, drain it of its resources, and then as soon as it's done, it's going to explode and it's going to release a bunch of viruses. So the viruses really don't care about destroying the genome. So it's going to break down the host cell's DNA. It's going to use those nucleotides. And it's going to start replicating its own genome. It's going to start replicating its capsids, all of those parts, and then it's going to start putting them together. So as the viral genome is replicated, each new virus's capsid is spontaneously assembled around its genetic information. So, we have that DNA, it turns into mRNA, and then these bacteriophages are going to use the host cell, that bacteria cell's own replication machinery, the ribosomes. It's going to use those to start making its own proteins. And as these capsid proteins and all these structural proteins that make up the bacteriophage are created, they spontaneously assemble around the genetic information. And it produces, a bunch of copies of that original bacteriophage. So as the number of newly assembled viruses increase in the host cell, the viral genomes start producing lysozyme. So that's another enzyme, but it's not an early gene. It's actually known as a late gene. The enzyme that will aid in the next step of the lytic cycle by destroying the bacterium's cell wall. So now that we're inside of the bacterial cell and we're creating more and more of these bacteriophages, remember the lytic cycle, at the end of it, the host cell dies. So at this point, these bacteriophages are trapped inside this rigid cell wall and they need a way to get out. So it's not enough that we're just creating more and more and more, because there's a chance that this cell might die and it'll never lyse. There are a couple of different ways that cells can die. So if that cell dies and everything inside it dies before these viruses are released, then the virus failed. It did not do its job. So what happens is these viruses encode for lysozyme as well. And as lysozyme is released, only after a certain amount of bacteriophages are created, it starts breaking down the cell wall. So once the cell wall is degraded, water continues to move into the cell. So that cell wall, it protects from outside pathogens, but it also functions to keep pressure equilibrium. So the osmotic pressure is pushing outward. As water moves into the cell, it's pushing outward, and that cell wall usually keeps the pressure inward, and it keeps that cell turgid. It keeps it very rigid. But when there's no more cell wall, water is going to continue to move into the cell, and it's going to continue to push out on the cell membrane. And the cell membrane does not do a great job of containing water. So without the cell wall counteracting that osmotic pressure, the bacteria cell bursts, it lyses, it dies, and it releases about 100 bacteriophages into the environment. So those 100 bacteriophages are then in the surrounding tissue. And if they're in a colony of bacteria, they can very quickly find another bacteria, and the process begins all over again. So they'll attach right to that wall, inject their DNA, and start this entire process over. And as I mentioned before, since lysozyme is created later in the infection, it is known as a late gene. And you can use this diagram to the left. It pretty much puts everything summarizes everything very well. So I added that if you want to like pause and take a look at it. Step 1, attachment, penetration, and then we're transcribing. Remember, bacteriophages carry DNA, so all they have to do is transcribe that DNA into mRNA and they're good to go. It's going to replicate, it's going to synthesize a bunch of proteins, assemble the proteins, and then boom, bursts after lysozyme. So while the lytic cycle is a very effective cycle that viruses use to spread pretty rapidly, it very quickly kills the host. So sometimes that's not really the best method. It's not really the best strategy for these viruses to spread. So remember that the viruses can't replicate without a host. So if a virus kills the host too quickly, it's not going to be able to spread. So some viruses enter what we call the lysogenic cycle. And this is a process in which the virus inserts its genome into the bacterium's genome. So it's a little bit more strategic and it's a little bit more insidious. So after the bacteriophage's genome is inserted into the host's genome, it's known as a prophage, and the cell is now known as a lysogen. So the prophage is not expressed due to a phage-encoded repressor protein. It remains silent and is reproduced as the host cell multiplies normally. So instead of coming into the cell, it enters the cell the same way. Say we're talking about a bacteriophage and it's entering a bacteria cell. Enters the same way. It injects the DNA in, and then it starts, it injects the DNA, and the DNA is in the cytoplasm. But instead of immediately starting to, break down the host genome and start creating more bacteriophages, what it does is it enters the cell's genome without destroying it, and it inserts itself into it. And it remains silent because of this repressor protein. So basically it stays in that genome silently. The cell is not aware that it's in the genome. It doesn't detect that anything's wrong. So the cell goes about its regular business, and what does this regular cell do? It replicates, it divides its own self. So as it's creating more, as it's multiplying, this bacteria cell is multiplying, it's also multiplying its own genome. And in that genome now we have that prophage. So it is inadvertently multiplying, it's inadvertently replicating this viral DNA, although it is still dormant. So eventually the cell switches from its lysogenic cycle to the lytic cycle after the prophage is removed from the host's genome in a process called excision. So the switch from the lysogenic to the lytic cycle can be caused by a number of factors, but it's usually the result of environmental stressors like hypoxia, toxins, stress. So an example, we're talking about bacteriophage, but animal viruses also undergo this lysogenic cycle. And one of the more common examples that you may see is the herpes simplex 1 virus, which affects humans. And that is the common cause for cold sores. So I think it's a vast majority of Americans, at least, do have this provirus inside their cells. It's just dormant. It's not expressed until there are stressful conditions. And for human beings, that could be actual stress. It could be starvation, dehydration, until something triggers that. And then we get an outburst because these cells that contain this provirus, this viral DNA, they switch from that lysogenic dormant cycle to the lytic cycle due to those stressors. And then that's how we get an outbreak. That's how those viruses start to spread. So that's why viruses can remain dormant for years before they're switched over to that lytic cycle. And if you look to the right, I put a diagram. That's the bacteriophage. It gives you sort of a good illustration of how the prophage is integrated into that circular bacterial genome. So the phage attaches to the cell, enters the DNA. Phage DNA enters the bacterial cell, like we said before. And then you see on that step 3, the phage DNA integrates into the bacterial cell. So you could see that prophage that's inside the bacteria, and the bacteria is not aware. It's not aware that there's a prophage inside its genome, and it's going to replicate. So you see it goes from one parent cell to 2 daughter cells, and both of those cells have identical genomes, including that prophage. And this can go on for a very long time until they switch to the lytic cycle.